Before we begin, are there any questions about the final exam? There are going to be 50 questions, all multiple choice, just like the last exam. The last exam, I think, had 40 questions, if I remember correctly.
Is it including the stuff that was
No, you guys...
Like from that last
Yeah, it's for you guys, for the pre-med 215, your finals are not cumulative. For my nursing students in 205, their finals are cumulative. So you guys, all you need to do is study the content from midterm all the way till today, okay?
Is this presentation our last grade before the...
Yeah, so yeah, the presentations are the last grade.
Do you know when they'll be inputted?
Sometime this week.
Okay.
Yeah, I finally caught up with grading last night. I didn't even though I was on vacation, I didn't really have much of a vacation. I was working almost every day. Anyways, all right, enough about me, about you guys here. So to start with, make sure you know your different augers, remember? So we started off with the McConkey auger when we were doing our unknown bacteria lab. Now, is a McConkey auger, well, let's start with the nutrient auger. Is a nutrient auger selective or differential? It's not, right? Anything is going to grow in a nutrient auger. So bacteria, as well as molds, fungi, will grow on nutrient augers. As opposed to McConkey augers, are they selective or differential?
They're both.
They're both, yeah, excellent. So they are selective and they are differential. What exactly is it that makes them selective?
Only selects gram negative.
Yeah, selecting for gram negatives, and it's going to have what? It has two things in there. Yeah, so it has crystal violet, and it also has bile salts, right? And so the bile salts especially are going to select for gram-negative organisms. Gram-negatives you often find in your GI tract, like E. coli, right? So a lot of the Enterobacteriaceae organisms are going to be found in your GI. So it's going to be selecting for those, and then it's going to differentiate how? Yeah, so lactose fermentation. So lactose fermenters are going to undergo a change in color, right? So it's going to look kind of what? Like kind of pinkish, right? It's going to look kind of fuchsia. And if it's like a serious lactose fermenter like E. coli, that fuchsia color is going to disseminate throughout the auger. So make sure you know what that looks like. If you see a picture of a McConkey auger, know what kind of organism that might be. Cool. So in that unknown lab, we were doing a bunch of series of tests, one of which was the catalase test. Do you guys remember what catalase does?
Breaks down hydrogen peroxide.
Yeah, so catalase is going to break down hydrogen peroxide into what two things?
Oxygen and water.
Oxygen and water. That's exactly why it starts bubbling, right? So if it's a catalase positive organism, it's going to bubble because of the release of oxygen in its gaseous form. Another auger that we used in the unknown bacteria lab was the mannitol salt auger. It was those of you that had gram positives, right? So the mannitol salt auger, what are the kind of organisms that are going to grow in there?
Halobacteria.
So gram positives, so it's like selective, right? And you're going to be able to differentiate between different types of gram positives. For those of you who used the mannitol salt, do you guys remember what it was? Like what happened? Did it change color at all? Yeah. Yep, exactly. So if it turns into like a yellowish color, you're thinking something maybe like Staphylococcus aureus. Think about Oro, right? Or AU is gold on the periodic table. Think about it that way, that it changes the mannitol salt into like kind of a goldish hue. We use another one. For those of you who had a gram negative, you guys used the other plate, which was the eosin methylene blue plate, which was the EMB plate. What happened on that? Certain types of organisms, well, first of all, gram negatives, right? And it's also differential, right? So it's selecting for gram negatives and it's differentiating between those gram negatives. Do you guys remember what happened? There was a couple that kind of looked like pinkish, purplish, but then there was ones that looked kind of green. Do you guys remember that? For the lab this semester, we were using Citrobacter fruendi. But I'm not testing on that one. I'm testing on the other bacteria that turns green. You guys remember which one that was? E. coli, yeah. So if you see greenish hue on your EMB plate, you're thinking something like E. coli. All right, let's talk about the bacterial genetics. When we did transformation of bacteria, we transformed the E. coli, and there was one thing. Let me see here. Hold on a second. On the medium, there was a beta-lactam drug, so it's ampicillin. Now, if it was able to grow on an ampicillin plate, what does that mean?
It's beta-lactam resistant.
Yes, it's resistant to beta-lactams. So that means it has beta-lactamase as part of its enzyme that allows it to be able to grow despite the presence of a beta-lactam drug like ampicillin. And then another thing that we used for one of those plates was a arabinose. Do you guys remember what a arabinose did?
It's the gene promoter.
Yeah, so it binds to the RSC component on the gene, right? So it inhibits that. So it allows for RNA polymerase to attach to the promoter region so that it can actually transcribe the green fluorescent protein. So if it was able to glow with GFP, that means that it was in the presence of arabinose. It needed arabinose to be able to do that. Let's see what else is important here. When we did the, make sure you know when it's green fluorescing. You make sure you know what contents are on there. It has to have the four things. So for that lab, we had ampicillin. It had the arabinose, right? That was the most important part, and it had pGLO, right? So the pGLO plasmid is what allowed for the glowing. In terms of the know that the glowing component was involving cyber green, okay? And then we put it underneath the UV light. And cyber green is what allowed for glowing, not for the pGLO, but for the, when we did PCR. Remember we did the PCR test and then we ran it through a gel electrophoresis. The reason why we were able to see the DNA is because of the presence of cyber green. Know what the definition of antibiotic is. Because there's chemotherapeutic agents, right? So things like salvarsan, protonsil, those kinds of drugs are going to be chemotherapeutic agents, right? They're chemicals. Versus an antibiotic. An antibiotic is specifically something derived from another living organism, mostly molds and bacteria, right? So think about penicillin, derived from penicillium. Streptomycin is derived from Streptomyces. So those are going to be considered antibiotics. In terms of testing antibiotics and their efficacy, what's the best way to do that?
Your inhibition zone.
Yeah, the zone of inhibition, what do you call that test?
Kirby-Bauer.
The Kirby-Bauer test, or it's also referred to as the disc diffusion test. Kind of similar to that, when we did our strep throat test a couple weeks ago, we put a little bacitracin tab on there. Some of you guys had different types of hemolysis patterns. What is it when you have partial hemolysis? You just see like a little bit of a clearing around the colony.
Alpha.
That's alpha hemolytic, right? What does it look like when it's totally hemolyzed and you can see right through it?
Beta.
That's beta hemolytic. So make sure you know the difference between those two. If there's no hemolysis and it just grows on there, that's a gamma hemolytic pattern.
What is the beta one again?
Beta means you can see right through it. It's total complete hemolysis of the blood agar. No hemolysis would be gamma hemolytic. Okay, moving on. We talked about catalase already. We talked about the zone of inhibition. So if you do see a zone of inhibition, what does that mean?
No.
It's the opposite. Yeah. Make sure you don't mess that up, because a lot of people mess that up. They think the zone of inhibition means that it confers resistance. It doesn't. You will see zero zone of inhibition if it's resistant, right? If you see a zone of inhibition, it means it's susceptible. Yeah, so culture and sensitivity testing, that means it's sensitive to the antimicrobial. We talked about the when we used a bacteriophage, right? when we were trying to do our PFUs, what are those little tiny stipples, those little holes that were punched into the colony? That's a plaque, right? So the plaque represents what? Just one single individual virus. One single virus can result in a plaque. Protozoans. So you guys did your infectious or pathogenic eukaryotes on your own last week. So what are protozoans? Are they prokaryotes? Are they eukaryotes? Or what are they? They're eukaryotic organisms, right? So they're eukaryotes. They can exist in multiple different phases, right? Some are cysts, some are trophozoites. If you see one that looks like a little teardrop shape and it's got two little eyes looking at you, it's got maybe like flagella, causes what they call beaver fever or traveler's diarrhea. What's that one called? No, that's Entamoeba histolytica. That doesn't have flagella. It moves through like pseudopods. But close. No. No. Plasmodium is a little bit different. No Giardia. If you see an image of Giardia, know what it looks like. And know if you see the two little eyes, it's a diplomonad, so it has two nuclei. If you see those two little eyes looking at you, that's going to be its trophozoite form. I'm not going to test you on the cyst form because it's a cyst. So know that they can exist in cystic form and trophozoite form. Speaking of treponema, Plasmodium, make sure you know how to identify the different forms of Plasmodium on a peripheral blood smear. Hint, the rings. Think about signet rings. I'm not going to really test you on the merozoites or the schizonts and all that. Mostly focusing on the rings. Know the signs and symptoms of Plasmodium infection. What are some of the signs and symptoms? I put a case presentation of a patient that I saw, and I had an image of what he looked like. Do you guys remember that? He was jaundiced. He had scleral icterus. Sclera icterus is yellowing of the eyes, of the conjunctiva, right? So that's going to be part of like the hepatic component to the Plasmodium life cycle. They're also going to have things like the signs of hemolysis, right? So tachycardia, tachypnea. They can also get splenic involvement, right? Because those damaged red blood cells get sequestered by the spleen. So.
Also liver failure, right?
Yep. You get liver involvement. That's why you get the jaundice. Know the organism that causes rheumatic fever. Do you guys remember? We talked about it quite a bit. Yes. Scarlet fever, which turns into rheumatic fever if left untreated. You guys remember that bacteria? Strep pyogenes, Streptococcus pyogenes, that causes strep throat. Causes lots of other things like necrotizing fasciitis, all sorts of other stuff. Toxic shock syndrome. But it's going to cause also, in some patients, scarlet fever, and then if left untreated, can turn into rheumatic fever, which causes what? What's like the main thing that is a huge concern with rheumatic fever? It causes infectious endocarditis, which causes, it affects one of the things in your heart. It's the number one cause of mitral valve stenosis. It's the number one cause of mitral valve stenosis. So make sure you know that. Know the different forms of the fungi. That was all part of that pathogenic eukaryotes. So there's things like sporangiophores, there's things like kinetophores. Know what they look like, okay? And then know the difference between Aspergillus, Penicillium, Rhizopus, okay? So know what those three look like on microscopy. If you were to see like a little image from a light microscope, be able to identify that.
What was that again?
There's three that you need to be familiar with: Aspergillus, Penicillium, and Rhizopus. Okay. In terms of fungi, know what the causative agent of valley fever. What causes valley fever? Do you guys remember? It starts with a C. Yeah, Coccidioides immitis, which would result in coccidiomycosis. Speaking of other fungi, Which is the fungus that causes oral thrush as well as like fungal vaginitis? Or like yeast infections? You guys remember that, or you guys know that from lecture, or from my presentation that I didn't teach you guys? I just had you guys look at. So candidiasis. So Candida albicans is going to be the one that causes oral thrush as well as yeast infections. One way to remember Candida is one of my students came up with this mnemonic. It's funny. The fun guy had candy. I don't know. I don't know if it works. Anyway, it's a common fungal infection, so you guys should definitely know that. There's going to be a couple different Helminths on your test. There's like the tapeworms. There's also the nematodes, right? What's like the main nematode that I showed you guys? It's like a smooth worm that looks like a bunch of spaghetti. It's kind of gross. I showed you guys a video of the intestinal obstruction with that one. Do you guys remember what it is?
Was it like the liver fluke?
No, not the liver fluke. No, the other one, Ascarius lumbricoides. So Ascarius, the way I like to remember it is like you have worms coming out of your Ascarius, your a-hole. So I don't know. It helps me remember. It's a really disgusting worm. So make sure you know the different worms. There's tapeworms as well. There's going to be Tinea solium as well as Tinea saginatum. So know what those look like. Okay.
The tapeworms are more associated with like raw fish or raw.
Well, there's a fish one, which is Diphyllobathrum blatum. I'm not testing you on that. But Tinea solium is pig tapeworm. Tinea saginata is going to be beef tapeworm. If you did get a Helminth infection, what kind of leukocyte would you see going up? Eosinophils? Yeah, excellent, because eosinophils are associated with parasitic infections. Let's see here. Know that going back to the virus plaques, what type of virus did we use? We used a bacteriophage, but what kind of bacteriophage was it? It was a T4. It has one cycle, right? It's a lytic phage, right? So the virulent T4 lytic phage versus a lambda lysogenic phage, right? The lysogenic phage is the temperate phage. It has two cycles. It has that lysogenic cycle, so it just continues to replicate in the bacteria. The bacteria continues to live its life, dividing and whatnot, and then eventually undergoes lysis. Let's see here. No ELISA, the antigens, right? So we're testing for antigens, right? With ELISA. Speaking of antibodies and antigens, know the region that's going to be associated with binding to the antibodies, right? Or epitopes. Sorry, not antibodies, antigens. So know the region. So there's two regions. There's the FC region, which is the crystallized constant region, and then there's the FAB, which is the antibody binding site, which is going to be the variable region. Know the things that antibodies can do, right? There's 6 different things that we talked about, but amongst them, there's going to be neutralization of toxins, right? It can also bind to flagella, causing immobilization. It can also cause opsonization. It can cause agglutination. So know all the different things that antibodies are capable of doing. Back to the bacterial genetics. For bacteria to undergo transformation, it has to be something. It starts with a C. Competent, yeah. So know what competency means. If you saw competence, know how to define it. Know the different steps in the PCR reaction. There's 3. What were they?
Annealing.
Well, the first one is going to be you're subjecting it to a high heat, so denaturation, then annealing, and then you have extension. And that repeats itself 30 times, and then you get, I think it's up to 1 billion molecules of DNA after those 3 steps. So heat was denaturation, right? So you're separating the nucleotide base pairs apart, and then you have annealing, and then you have extension. Know the charges of your DNA and why that contributes to, or how that's useful in gel electrophoresis. Know how to calculate your plaque forming units. It's literally, I'm giving you the equation. Just know how to move the decimal points, right? Know that 10 to the negative 1 means 0.1. 10 to the negative 2 is 0.01. So just know how to move the decimal points when you divide. Know what dimorphism is in mycology, right? So a lot of those, like for instance, Coccidioides immitis, it can be a spore, but they can also become hyphenated, right? And then mycelia. So know what dimorphism is. And then definitely know rheumatic fever. We already talked about that with strep pyogenes. And know what kind of tests you might want to do in order to be able to actually diagnose and cure and treat strep throat. That is it. Any questions? All right, guys. Right on. That was longer than I was hoping. It was 20 minutes. I was hoping 15, but...